Welcome to Zoology class. So in the last class, we have discussed Frederick Griffith's experiment on mice using Streptococcus pneumonia bacterium to identify which is the genetic material, to ascertain whether it is the DNA or the protein, the genetic material. And at the end of his experiment, even though he could not prove which was the genetic material, he could identify that is the material that can transform one cell to another, or that is responsible for bacterial transformation. So by that experiment, it was very clear that there are certain molecules or substances in the cell that determines the properties of a cell, or when transferred from one cell to another cell, the cell to which it is transferred the properties can be modified. And later, the experiments, the modified experiments conducted by 3 scientists proved very clearly that DNA is the genetic material. It is not the protein, but the DNA, the genetic material. And in 1952, 2 scientists, Alfred Hershey and Martha Chase, they conducted a very interesting experiment, a more genuine experiment, by using E.Coli bacteria and bacteriophage viruses to prove beyond doubt the molecular nature of genetic material. Or to ascertain without doubt the nature of the genetic material. So they conducted the experiment in the year 1952 by using E. coli bacteria and bacteriophage viruses. So let us look into the experiment conducted by Alfred Hershey and Martha Chase. They cultured the bacteriophage. You know what is bacteriophage? Bacteriophages are viruses that attack bacteria, or viruses which use bacteria as their hosts to multiply. They are called bacteriophages, or simply phages. Okay. So Hershey and Chase, Alfred Hershey and Martha Chase, they cultured these bacteriophage viruses in 2 culture media. Medium A and medium B. In medium A, they added radioactive sulfur, or they made radioactive sulfur available in the culture medium A, where some bacteriophages were grown, and they introduced radioactive phosphorus into culture medium B, where some bacteriophages were grown. These viruses multiplied in these two media by using the nutrients provided in the culture medium. So, for bacteriophages or viruses in the culture medium A, radioactive sulfur was available apart from other elements or other molecules required for their multiplication. And in culture medium B, radioactive phosphorus was available for them apart from other essential elements for their multiplication. Sulfur generally is a part of protein. And phosphorus is a part of DNA. So those viruses which were grown in the culture medium A took up the radioactive sulfur to construct their protein coat. Viruses have a protein coat and a genetic material inside the protein coat. So the genetic material may be DNA or RNA in viruses. So here, a DNA, bacteriophages used were DNA viruses. The genetic material was DNA. And those bacteriophages grown in the culture medium A where radioactive sulfur was available, took up the radioactive sulfur to construct their protein coat. So, radioactive sulfur became a part of their protein coat. Because sulfur is a part of protein. It's a component of protein. And those bacteriophages grown in the culture medium B took up the radioactive phosphorus, because that was available there, radioactive phosphorus, to construct their genetic material DNA, and their DNA or genetic material contained radioactive phosphorus, because phosphorus is a part of DNA. Okay. Now these two bacteriophages grown in separate culture media, into these culture media A and B, they allowed or they introduced E.Coli bacteria to both of these culture media. And what would happen? These bacteriophages in both the media would attack the bacteria introduced into the culture media. And what is the mode of attack of a virus on a host cell? A virus, when attacks a host cell, it leaves the protein coat outside the host cell, and only the genetic material will be introduced into the host cell. And after entering into the host cell, this genetic material would take up the command or take over the command of the cell, and by using the machinery of the molecular machinery of the host cell, they produce copies of the DNA, and viral particles and several viruses would be formed, and by breaking the host cell they would be released outside to attack fresh host cells. So this is a mode of attack of a virus. So the same thing would happen here also. When E. coli bacteria were introduced into these two culture media where bacteriophages are grown, these bacteriophages attack this E. coli bacteria in the usual manner. They left behind the protein coat outside, and only the genetic material was introduced into the host cell. You know, what would be there in the culture media now? The protein coat of the virus would be left outside the bacteria, and the genetic material is inside the bacteria. So after some period of time, both culture media were subject to centrifugation. Centrifugation is a process where substances are separated from a mixture of substances based on their density, their size, etc. So when these two cultural media were subjected to centrifugation, centrifugation is a process in which a liquid substance is allowed to spin at a high speed. When it is spun at a high speed, substances would get separated based on the density. The denser substances, the denser particles or denser molecules would settle down, and the lighter substances would float on the surface. It will become so the settled part is called the sediment, and the upper portion where the light particles contain is called the supernatant. So after the centrifugation, the culture media would be separated into two parts. The sediment, where heavier or denser particles are settled, and the supernatant, where lighter particles come. And after the centrifugation, both these components, the sediment and the supernatant of both the media, both the cultural media, were subjected to radiation detection. And it was identified, or it was understood, that in culture medium A, radioactivity was observed in the supernatant, and in cultural medium B, the radioactivity was identified in the sediment. What does it indicate? In cultural medium A, the radioactive substance was sulfur. Sulfur, I told you, is a part of the protein. So, bacteriophages took up the sulfur to make their protein coat, and while attacking the bacteria, they left the protein behind that cannot go into the cell, into the bacteria. Only the genetic material can go into the bacteria. And this protein coat is lighter compared to the bacteria, and they appeared in the supernatant, not in the sediment. And in the culture medium B, the radioactivity was detected in the sediment, not in the supernatant. Here, the radioactivity is due to radioactive phosphorus. In the cultural medium, there was radioactive phosphor. So that radioactivity is due to radioactive phosphor, and phosphorus is a part of the DNA. So the reason for the radioactivity in the sediment is due to the presence of the radioactive phosphorus in the DNA that went into the bacterial cell. In the culture medium A, the radioactive sulfur was a part of the protein, and there the radioactivity was detected in the supernatant. That means that protein did not go into the body, into the cell of the bacteria. It remained outside. And in the culture medium B, radioactivity was detected in the sediment, because radioactive phosphor, in the culture medium B, there was radioactive phosphor, and this radioactive phosphor became a part of the DNA, and that DNA entered into the bacteria. So from this experiment of Hershey and Chase, Alfred Hershey and Martha Chase, it was proved beyond doubt that it is the DNA that is the genetic material, not the protein. As it was the DNA the genetic material, viruses introduced DNA into the bacterial cell. If protein was the genetic material, viruses should have introduced the protein into the cell. It was made very clear that DNA is the genetic material, not the protein. So it was established that DNA is the genetic material, not the protein. Okay. Now, further investigations, further researches revealed that RNA can also act as a genetic material in some cases. So DNA can be the genetic material, and DNA is the genetic material in majority of the cases, but RNA can also be the genetic material in some cases, especially in some viruses. They are called RNA viruses or retroviruses. Okay. Now let us see what should be the quality of a genetic material. What are the properties of a genetic material? The genetic material should have certain unique properties. One of the properties of genetic material is that it should be able to generate its replica. That means a genetic material should be able to replicate or produce its own copy. It should produce its own copy. So when we examine whether this property is right for DNA and RNA, both are genetic materials, we can understand that both the DNA and RNA can replicate. They can make their copies. So both DNA and RNA pass this criteria or pass this qualification. The second one is that genetic material should chemically and structurally be stable. The genetic material should structurally and chemically be stable. So there should be stable. Let us see whether they are stable. DNA and RNA, how they are stable and to what extent they are stable. The stability of DNA was proved by Griffith's experiment itself. When Frederick Griffith heat killed the smooth strain, the bacteria died, but the DNA was intact. The high temperature or the heat could not destroy DNA. That we have already seen. So it is stable, physically it is stable. And DNA is very less reactive. Usually, it does not undergo any chemical reaction. So it is chemically also stable. RNA is also stable, but not to the extent of DNA. RNA is less stable than DNA because of certain reasons. Why is RNA less stable, chemically less stable than DNA? There are certain reasons for that. One of the reasons for less stability of RNA is that RNA, the sugar in RNA is ribose sugar. And the sugar in DNA is deoxyribose sugar. And in ribose sugar, one oxygen atom or one OH group is extra, one OH group is excess than in the deoxyribose sugar. And this OH group involves in chemical reactions, and thereby an RNA is more reactive than DNA. So that gives this property of the RNA, provides it less stability than DNA. So the addition, this OH group, one extra OH group present in RNA, makes it less stable. This is one reason. And another one is that in RNA, uracil is present. We have studied the structure nucleotides in first year. So nitrogen bases are there. One of the nitrogen bases in RNA is uracil, and this uracil is not seen in the case of DNA. Instead of uracil, the nitrogen base is thymine. And the uracil is more reactive than thymine. So presence of uracil in RNA makes RNA more reactive and less stable than DNA. And another is RNA sometimes acts as an enzyme. That enzyme is called ribozyme. This is present in the ribosomes. An enzyme ribozyme is an RNA and it acts as an enzyme. So of course it is reactive. Okay. So when we compare about the stability of RNA, compare the stability of RNA and DNA, even though both are genetic materials, we can say that DNA is a better genetic material as far as stability is concerned. RNA is less stable, so it is not a good genetic material, or it is not a better genetic material. As far as stability is concerned, chemical stability is concerned, DNA is a better candidate or better genetic material. Okay. Now let us come to the next property of the genetic material. A genetic material should undergo or should be subjected to mutation. should be subject to mutation. So both RNA and DNA are mutable. Both are subject to mutation. But here also, RNA is more mutable than DNA. It gets mutated at a faster rate. That's why RNA viruses in which RNA is the genetic material, mutates faster and new forms are formed at a faster rate. The best example is coronavirus. It's an RNA virus. The genetic material is RNA. And now we understand that this coronavirus mutates at a faster rate and different varieties of this virus have evolved, have formed, and as a result of this, the symptoms and the seriousness of this illness varies from area to area or person to person, because it undergoes mutation and so many varieties have already evolved, because it's an RNA virus and RNA gets mutated at a faster rate. But DNA mutates very slowly. Okay. Then another is, a genetic material should be able to express the characters. A genetic material should be able to express phenotypic characters. Should be able to produce the phenotypic characters. DNA, of course, is capable of producing the phenotypic characters. RNA can also express the phenotypic characters. But here, DNA requires RNA for phenotypic expression. Or DNA cannot directly express the phenotypic characters. It expresses the phenotypic character through RNA. So as far as expression of the phenotypic character is concerned, RNA is better than DNA. Because DNA alone cannot express phenotypic character. The phenotypic character is expressed by synthesizing proteins. RNA is a part of the protein synthetic machinery. DNA determines, of course, it determines or it decides what type of protein should be synthesized in the cell, but that is carried out only through RNA. So RNA directly involves in the protein synthesis, thereby the expression of the phenotype. And DNA is a better genetic material for storage of information. For storage of information. So when we compare the DNA and RNA, we can understand that DNA is a better genetic material than RNA. Mainly because DNA is more stable and less mutable. We shall end up the class here. We shall see in the next session.